Thank you for the opportunity to speak to this wonderful audience and to speak after these fantastic researchers whose work I've been following for many years and very inspired by their work. A few disclosures for some collaborations, advisory boards, review panels, and editorial boards, as well as our funding support. The roadmap for my translational talk is shown here. I'll talk a little bit about the gene by environment interactions that happen in Parkinson's and how we feel they converge on inflammation. Something about immune aging and the role of inflammation in chronic systemic disease in brain inflammation. The innate and adaptive immune contributions in Parkinson's. How we feel that the perfect storm occurs in Parkinson's. Okay, that did not just happen. Are you still watching? Okay, great. The non-motor features of PD, which have been alluded to earlier, and how the GI dysfunction and dysbiosis in Parkinson's has a role in immune dysfunction as well. We'll talk about one of the genetic contributors to Parkinson's, and that's LRRK2, and how that might be the missing link between inflammatory bowel disease and Parkinson's. And then how the gut-brain neuroimmune dysfunction may contribute to PD and implications for therapeutic strategies. So I'll start with this schematic from a recent review in movement disorders from our group in collaboration with Christine Klein and Andy West, where we talk a little bit about how environmental factors, which we know have been implicated in Parkinson's, may disrupt immune function, alteration of adaptive and immune cells through various mechanisms that cause tissue damage, and in collaboration with other things may trigger systemic inflammation, which contributes to neuroinflammation and increases the chances that you will develop Parkinson's disease. So I'll refer you to that. And then I'll step back and tell you that we know that bidirectional communication between the nervous system and the immune systems in the periphery is critical for brain health, using this schematic from my colleague Tony Wascore, knowing that at the cellular, at the system, at the tissue level, there is communication using cytokines, which not all of their functions are well known, but we do know that they play important roles in communication between different cell types. And when there is imbalance at these levels, it will set you up for various pathologies. Now we know that the brain-resident innate immune cells of the brain, the microglia, are very important for a lot of different functions, including trophic support, phagocytosis, cytokine secretion, and they move around and that they survey the environment. And one of the important roles of these microglia and brain-resident immune cells is to perform something called antigen presentation. And this leads to the maturation of the adaptive immune cells in the body and the brain. And that gives rise to mature T cells that differentiate and take on very specialized roles depending on their environment. And this happens because innate immune cells process various peptides or pathogens that they engulf, and they put them on their surfaces. They decorate themselves using MHC class 1 or 2 receptors that they then present to naive T cells to cause them to differentiate and mature into various specialized subtypes. Now, one of the things that has happened is because of infectious disease, there has been evolutionary pressure in humans for the development of genes and gene variants to give robust immune responses. But because we've lived too long, there has been chronic antigenic load, and through a process of inflammaging, which is composed of low-grade chronic inflammation and immunosenescence, which I'll describe in the next slide, there's been loss of immunocompetence and autoimmunity, which we and others believe contributes to age-related disorders. And keep in mind that age is the #1 risk factor for AD and PD. So what is immune aging? We know that it has two heads, and Jay Garanzi and colleagues have described it very well in humans. It's an age-related immunodeficiency described here, where there's a shrinking of naive T and B cell compartments, contraction in T and B cell receptor diversity, and a decreased sensitivity to any of the stimuli that activate them. On the other hand, there's an age-related inflammatory syndrome where there's the preponderance of the myeloid cell lineages take over the lymphoid lineages. And then there's excess production of inflammatory cytokines, and there's a failure of self-tolerance with the production of autoantibodies. So together, you end up with an impaired adaptive immune response, a constitutive low-grade inflammation, and autoimmunity as you're aging. And what we believe is an important context is that all these age-related disorders of neurodegeneration, they're proteinopathies, right? And so if you have an excess production of amyloid or synuclein or Huntington's disease or oxidized protein, because you have a mutation, it's easy to see how you might trigger microglial activation. And that has been shown in vitro and in vivo and in humans with these mutations. And then you set up a cascade of inflammation, apoptotic cell death, and all these diseases. And that's a $64 million question. How do you have these divergent triggers converge on inflammation and then diverge again, right? But what about the 95% or so of individuals that develop the sporadic diseases and don't have these mutations that would set off this cascade? So we and others propose that you can still get to these cascades through other means by activating the chronic inflammatory cascades through unhealthy aging, chronic systemic disease, inflammatory diseases like obesity, maybe atherosclerosis, type 2 diabetes, and environmental exposures, potentially peripheral infections, pesticides, physical inactivity, unhealthy diet, and dysbiosis. And this is just a model that has been tested in many labs, including my own, to try to link chronic inflammation to neuroinflammation and neurodegenerative disease. So if perhaps innate immune dysfunction that occurs through this chronic inflammation, because we believe that the chronic inflammatory syndromes probably cause the innate immune cells to become dysfunctional themselves. As you're aging, it's possible that then you have additional factors like stress and lifestyle choices, environment, diet, that then compound these potentially dysfunctional pathways that may be occurring, potentially that are genetically set in, right, through some of these variants that we are learning about, that I'll talk a little bit about and that Dr. Shapira mentioned. And they converge and they contribute to an acceleration. So you have environmental and genetic factors that contribute, and then you now must consider the innate and adaptive immune function in the periphery, and then potentially the gut. So the immune system is the perfect arbiter of the gene by environment interaction that you're seeing in PD and potentially other diseases. Now, is there evidence for this? So I don't really have time to tell you a lot about it, but I want to point out a few papers that support this. We know from animal models, one of the things that has been reported by Ashley Harms, a former trainee of ours, then in the laboratory of Dave Standard, and now on her own at the University of Alabama in Birmingham, is that peripheral monocyte entries required for alpha-synuclein-induced inflammation in the synuclein model. And we also know from the work of Dave Sulzer that they've identified specific T cells that may be seeing synuclein peptides, and that these T cells may be seeing them in a specific time in the early PD stages, and they persist and may correlate potentially small sample size, but needs to be replicated with early clinical stages. And so there is starting to be some potential support for the innate and adaptive immune system in pathway dysfunction in PD. So if you start thinking about motor and non-motor symptoms for PD, how can we put together, say, a timeline of when this occurs? So let's say you're born with so many dopaminergic neurons and you have to cross that threshold to develop the motor symptoms. And then, before that, you have some non-motor symptoms. And is the immune system playing a role before that, during that, or after? So when exactly should we be paying attention to the role of the immune system? And what we believe is that the perfect storm for Parkinson's development probably occurs early, that the immune cell function that I described, the immunosenescence, is happening early, is happening in the pre-motor early stage, 1 to 2 decades before you actually develop. So you have immune cell aging, and then you have the gene by environment. And so the perfect storm is happening here. And so we need to be paying attention for biomarker development here to identify people at risk with gene by environment interactions. And then, of course, we can use it to monitor early stages during the non-motor symptoms. And then, of course, try to figure out how to do biomarker development throughout for clinical trials. So what are those non-motor symptoms that we could monitor? GI dysfunction is a perfect one. We know that chronic inflammation is probably a driver in a multisystem dysfunction, and we have been paying attention to tumor necrosis factor for many years. We know that dysbiosis has been reported by multiple groups, including Payami's group, where they implicate a lot of different pathways in dysfunction in various populations. We collaborated with them, and we were more interested in what about the host? Is the host inflamed? Do they show signs of inflammation in their stool, in their gut? And so Madeline Hauser, a student in the lab, looked at the stool cytokines and found 4 of them were different in the PD patients versus the spouses. And you have to be very careful because you can't really compare PD versus no PD. You should be comparing someone that lives in the household as a real control. And so we found 4 of them that were very different between the 2 of them, and they're all NF-kappa B driven. We also found fetal calprotectin and lactoferrin. And we found definite incidence of digestive issues, anxiety, depression, and incidence of IBD and Crohn's, which was twice as much with them than with the spouses. And then we replicated this recently in a different cohort with Phil Sheperyans in Finland. And we found some really interesting things there too. In this case, we had the plasma to compare it to. And we found, again, a small cohort, but we found that females with PD had a higher prevalence of gut inflammation. And we found that males with PD versus those with no PD had lower short-chain fatty acids, and that PD patients with gut inflammation started showing disease symptoms at an earlier age, while patients with higher short-chain fatty acid levels were older when their symptoms appeared. And then we found certain kinds of bacteria might have affected the PD patients differently than the healthy controls. So these are preliminary findings in terms of size of cohorts, but we think that it's important to recognize that the inflammation that's happening in the gut is not reflected in the plasma. So while we all want to find signs of inflammation in the plasma to monitor them in clinical trials and see what's going on, it may be very local, and we may need to pay attention to other places, not just the plasma. So while intestinal inflammation may drive PD pathogenesis, what is the mechanism? And so we want to know what is the possibility that we can link the genetics to the gut inflammation. And I think Tony Shapira brought up LRRK2. It sits in the 3 buckets of high risk, low risk, and intermediate risk. And what I want to point out that in the very common variants that confer low risk, there are a lot of immune gene hits. And this is where we think you are going to have the interplay with the environment. And so if you look at LRRK2, it sits in the 3 buckets, and it is highly expressed in immune cells. And so again, when we look at immune cells in the periphery, monocytes and cells in the gut express a lot of LRRK2. And we know that there's been an implication of inflammatory bowel disease with incidence of Parkinson's disease, especially in Ashkenazi Jew populations. And so in a small study, again, by Inga Peter, she reported that patients with inflammatory bowel disease are more likely to develop PD. And then very interestingly, that those patients with IBD and anti-TNF therapy had a reduced incidence of of PD by 78%. So that's, very, very interesting to see. So is it possible then that some kind of intestinal inflammation and some kind of anti-inflammatory, anti-TNF therapy could reduce the risk? I think it's probably worth looking at. And then what about the LRRK2 association? Is LRRK2 that missing link between the gut and the brain and IBD and PD? And so this is from a recent review that's in press now in Nature Parkinson's disease, where we suggest the idea that LRRK2 may be the link between Crohn's and Parkinson's disease, because there are genetic variants that increase your risk for Crohn's. And we've also reported that in sporadic Parkinson's disease, individuals with Parkinson's have increased levels of LRRK2 in all their immune cell subsets. And these immune cell subsets are overproducing inflammatory cytokines and may be somewhat dysregulated. Now, we don't know the reason why they're overproducing LRRK2 protein. It may be because they're trying to curb inflammation. So I'm not suggesting that it's a bad thing. I'm suggesting that it may be an adaptive response to something that's going on in the disease. Now, why does gut inflammation matter? Well, Sampson and Sarkis-Mesmanian suggested that gut microbiota was important in regulating motor deficits in a mouse model of Parkinson's disease. And Roger Lytle suggests that if you have dysbiosis in the gut, it could trigger synuclein increases, and that could in turn pass on some kind of modified synuclein to the enteric ganglia, and that could then propagate up towards the brain. So that's the anatomical link, right? Gut inflammation and that potential propagation, and I say potential because it really hasn't been shown in humans, but it has been shown in animal models. So why do we care at all? Well, John Cryan and his group have shown beautifully that the gut microbiota can influence peripheral immune cell traffic for other potential diseases of the brain, such as schizophrenia. And so apparently if you disrupt the microbiota, you can affect what's coming out of the bone marrow. You basically affect the epigenetic reprogramming of these cells, and you affect what's going on in the brain. So we may have an opportunity here to target microbiota, to target the peripheral immunity in multiple neurological disorders, not just Parkinson's disease, by affecting the gut-brain peripheral immune system conversation, not just for PD, but for multiple brain disorders. And I want to put in a plug for the Mind Mood Microbes meeting that's going on virtually in May, where they'll talk about all these things with fantastic speakers, including Cryan and Elaine Chow, who came from that work. And I want to finish with this three-way schematic, which kind of alludes back to my first slide, with that bidirectional communication between brain and peripheral immune system. We now have a third player, which is the gut. And so the CNS-Gut-Peripheral immune system is a three-way axis, which is very complicated. We don't really understand it, but we know that by understanding the functional relationship between these three locations, we will be able to identify not just the basic function of these individual molecules that are coming from each of these sites and how they influence the function of the other. And we'll be able to identify targets for intervention to be able to prevent and potentially slow down the development of these diseases. And with that, I want to just conclude this bidirectional communication is really key for brain health, immune cell aging, and chronic inflammation resulting from the gene by-environment interplay represent the perfect storm for PD development. And in humans, the lifestyle choices, diet, exercise, all affect the G by E interplay and will impact gut and BBB permeability. Mitigating this chronic inflammation may reduce the innate immune dysfunction and risk for PD development and progression. And as I said, this understanding of the functional relationship between this axis may identify new targets for intervention. So the work started at Emory. We're now at University of Florida at the Norman Fixell Institute. And happy to take any questions.
Thank you very much. That's a fascinating area. If there are any questions, please do everyone put them in the Ask a Question section. If I could perhaps just ask one question. So you referred to the early phase of Parkinson's and the prodromal phase. Do you think inflammation is still a factor in the later stages of the disease when we still have progression, or do you think it no longer is an important factor at that stage?
Yeah, no, I think that's definitely there. The earliest sign of inflammation was postmortem brains by McGeer and colleagues. We know that microglia that are activated remain in the substantia nigra and the striatum, that there's astrogliosis. It's hard to understand, hard to know what the function is there at the end. I think at that point, it may be that they're there to, clean up dying neurons. I think the role early on of the innate and adaptive immune system is to help what's, a dysfunctional neuron. I think the role of the immune system at various stages may be very different. So I do think that inflammation probably starts early. At least that's what the PET imaging suggests. The PET imaging isn't perfect either, but I do think that what we know from the epidemiology, the PET imaging, the biomarker, biofluids, and the postmortem histological studies is that it starts early, it persists, and it probably has different roles throughout.
So more a reaction rather than causative later on, perhaps.
Maybe, yes. I think it may be a response, an adaptive response, and then later it may be driving, some of the cell death. Absolutely, yeah.
And one more question. We clinically we see a lot of different subtypes and different rates of progression and different probably prodromes as well. And do you think that do you have a hunch or perhaps even data whether inflammation may be more important in some of these? And you've referred to the gut pathology a lot, and that clearly is very important. But perhaps other forms like RBD, do you think it might play less of a role there?
Yeah, that's a great question. I think that Duda and Ron Postuma have, some really interesting data and have suggested that there is some evidence that patients with RBD may be more inflamed than others. And I was listening to a seminar the other day where Ron Postuma suggested that would be the patients that they would go into for a clinical trial to look at immunomodulatory and anti-inflammatories, because you can monitor progression and clinical changes and look at their scans and look at DAT scans and other things. So I think that would be very exciting, and I would be super happy to be part of that and tell them what to look for and tell them what to monitor. I think that would be a tractable group that could be monitored with various things. And maybe you could do both. You could do monitor their peripheral signs and their central compartment. Yeah.
Thank you very much.